Good morning. We don't have everyone who's registered here yet, but we're going to start because our speaker has to leave around 11:45 today, and I think we better get him started. I'm Millard Sussman. I'm a member of the Electronic Technology Committee, which organized today's lecture. Let me say just a word about why the Electronic Technology Committee is sponsoring a lecture on a topic that seems sort of remote to electronic technology. We have in the past sponsored some very popular sessions on genealogy, sessions in which we discussed internet tools for scrounging up facts about your ancestors. And today's seminar deals with the use of DNA sequencing to learn about our forebears and ourselves. The subject seems a natural connection for people who are interested in genealogy. There's a lot of technology involved in this, although most of it isn't the kind of technology that you're going to be able to use in your own hands. But we can do things that we couldn't imagine doing some years ago. I had a brother-in-law, for example, who I'm sure was 50% Neanderthal. I would love to see his report from Ancestry. So let me introduce today's speaker, who is standing next to me, Jason Fletcher, who's a faculty member in the La Follette School of Public Affairs. He came to us in 2016 as one of a cluster hire, focusing on a university initiative in social genomics. The faculty cluster hired, that is the people in the cluster hired in the initiative, include an interdisciplinary group of 4 faculty members holding appointments in sociology, public affairs, population health sciences, psychology, and biostatistics and medical informatics. And Jason is in himself a study in interdisciplinarity. He has a BS degree in economics and public administration from the University of Tennessee in Knoxville. He earned MS and PhD degrees here at the University of Wisconsin in applied economics. He subsequently taught on the faculty of Yale for a number of years. Spent 2 years as a Robert Wood Johnson Foundation Scholar at Columbia University. He had a Career Development Award from the W.T. Grant Foundation to study the interaction of genetics and social setting on youth development. His background is decidedly interdisciplinary. And his appointments are, he's appointed everywhere. You rarely encounter somebody. He's a professor of public affairs and sociology, director of the Center for Demography of Health and Aging. He's the executive director of the Wisconsin Federal Statistic Research Data Center. He's affiliated with the Department of Agricultural and Applied Economics, the Department of Population and Health Sciences, the Center for Demography and Ecology, the Institute for Poverty, and the Center for Demography and Health and Aging. I used to think with 2 appointments in departments that I was appointed in a lot of departments, it seemed like a lot of executive committee meetings to go to. You can imagine what Jason's life is like. Along with Dalton Conley, he's the author of a book, which is sitting here, The Genome Factor, What the Social Genomics Revolution Reveals About Ourselves, Our History, and Our Future. And it is a pleasure. I hope you will join me in welcoming Jason Fletcher, who will tell us what our DNA can tell us about ourselves, our history, and our future.
Thank you. Thanks for that very warm introduction, and thank you all for being here. I want to first thank, so part of my public talk tour is sponsored by a very generous gift from Senator Herb Kohl to the La Follette School of Public Affairs. So I've been going around the state with several of the Wisconsin Alumni Association have the Badger Cafe series. So I've done that a couple times, and we'll do that in Milwaukee, I think, in the next 6 months. But this is all to, the gift was in furtherance of the Wisconsin idea, which is to take the knowledge that's generated in the university system to all parts of the state and outside the state for that matter. So I want to recognize Senator Herb Kohl's generous gift and the La Follette School in supporting this. And like you saw, so I have a little bit of background about this book that I'll leave a copy here for anyone who's interested in reading it. That's why I brought one of the new paperback versions. So I wrote the book with Dalton in between fall 2014, fall 2016, and then it was published about almost 2 years ago, about January 2017. And the purpose of the book is to catalog some of the past attempts to integrate genetic data into social science research. And then what struck me, though, now that it's almost 2 years old, is how old it is, how many technological innovations has happened just in the last year and a half or 2 years. So that's point one, which is that this is a historical book already, even it was published a year and a half ago. And the other point is that we're woefully behind as a society in thinking about what policies we want to put forth in dealing with what we're calling the social genomics revolution. So that's what I'm going to be talking about today. And I'm happy to field questions both during the talk, and I'll try to repeat them so that they're recorded, and also we'll have a set time of Q&A after the talk. So please feel free to interrupt throughout. So let me claim that there's been a genomics revolution. Let me build that argument a little bit. So to have a social genomics revolution, I think we need a genomics revolution. So let me describe what I mean by that. So I'll first do that by describing a couple facts to get us all situated in what I mean by the genomics revolution. So first fact is to think about the human genome itself, which is complicated. There are approximately 3 billion pairs, I'm going to call them letters today, but they're nucleotides that have, in each location across the genome, you'll either have an A, a C, a T, or a G all the way throughout. So you're probably familiar with this spiral staircase of A's and C's and T's and G's. There's about 1.5 billion miles of DNA in your body. That's about 3,000 round trips to the moon. If each of these letters, if each of these nucleotides was a letter of a textbook, you could stack the pages 65 feet high. It's a complicated and rich source of information in the human body. Probably more than 99.5% of every one of these letters is identical across every human who's ever lived. So there's a common humanity that's written in these letters that we're so similar. So that's point one, similarity. Point two is that we are also 98% of these letters are exactly the same with chimpanzees. That, I think, suggests that some of the letters matter a lot. Just a few changes, 1 or 2% of difference across these 3 billion letters are the difference between us and chimpanzees. So that's the second point. The differences can matter. I have one example here, which is to read a small segment of the human genome in two different people. And these are identical, so you'll see the letters I just described, some C's and A's and T's and G's, C's and A's and T's and G's. And what this slide is describing is that there's one difference of letters in this genome. There's a T here, and if you come down here, there's an A. Otherwise, this whole line of letters is identical. But that difference, that single change in the genome, is the difference between having normal blood cells and sickle cells. So that's a point that some of these letters really matter. Having one letter versus the other can really matter for your life circumstances. there's only a small number of letters that, if you read through your genome, would be able to indicate whether you are lactose tolerant or not. Very small. And similarly, there's a very small set of letters that are determining if you have blue eyes. So there's some letters that really are easy to see how they make a difference. With that, they make a difference. The other point is that these letters are permanent. You've had these letters in your cells from the moment that you were an embryo. You will have the same letters in your cells approximately. I have a little asterisk here because I want to fend off your questions of saying, well, there's a little bit of this, a little bit of that, there's mutations throughout your life, that's all true. But to a first approximation, all those 3 billion letters you've had your whole life, and you will have the rest of your life. And the other part of the asterisk is anticipating potential questions about gene editing, which we might get into later. So that would be another instance where you have letters that differ now than when you were born. But, again, first approximation, all the letters that you have, you've always have had, and you always will have. Okay, so this isn't revolutionary. I'm describing some facts. Okay, so let me move a little bit closer into what was revolutionary here. So I've set up the facts. Those aren't revolutionary. What are technological improvements in actually measuring these facts. So I'm going to spend a few moments on what those improvements are. And then I'm going to add improvements in a different part of our world in statistical analysis that work together with the technological improvements to then generate my main point, which is that there are many new policy questions that we are a decade behind of even considering. So what are these improvements? Let me make a few short claims about the level of improvement. So this slide has nothing to do with genetics. This is to just remind ourselves that prices change over time. If we centered ourselves 20 years ago at the price level of college tuition and fees, what this yellow line is saying is that in the last 20 years, college tuition and fees have gone up by a lot. That's not news to probably many of you. The expenditures on education, or the price of just general education, has gone up. The prices of child care have gone up, have doubled in the last 20 years. Medical care prices have doubled. These are in our normal range of what we've lived through. In the last 20 years, prices have gone up in some instances, like college tuition fees, child care. But in some parts of our life, they've actually gone down by a lot. So TVs now are a lot cheaper for what you're getting than they were 20 years ago. And what I want to describe here is just this is the normal parts of our life. Prices go up, technologies change, prices go down, TVs are cheaper than they used to be. Okay, so let me take this normal change and then describe what has been happening in the cost of sequencing human DNA. So let me first gauge what I'm about to say by saying that in our recent memory, the fastest reductions in costs in our world have been about computing speed. And that's described in Moore's Law, where the idea is that every 18 months to 2 years, we have approximately a doubling of the power we get for the same cost. And so that's very fast. Every 18 months, double, that's very fast. So that's the slow line here. What the green line is describing is the cost per sequencing human DNA over the last 15 to 20 years. And that has fallen by a factor of 100 million. There's no precedent for that we have in any written records that over 15 years the cost of that technological advances have led to the cost reductions of that magnitude, 100 million times. So 15 years ago, when the first human genomes were being sequenced, it cost in the neighborhood of $3 billion. Now they're well below $1,000. In the very next 6 months to a year, I think people are thinking they'll be well below $500, well below $100 for human DNA sequences. So going from $3 billion to $50 over 20 years is unprecedented. So that's going to have some implications. This is just an example from last year saying, is this company really going to sequence for $80? That's what's on the table, not thousands of dollars, but $80. This is one of the best technological improvements from a year ago, which is, I have my laptop here. For $1,000, I can have a portable human DNA sequencer. So when Miller was saying we don't have that here, we could. I could have brought a $1,000 device and started sequencing all your genomes. So, and this is now going to tie to the introduction a little bit more fully, is that because of how cheap this is, there's been a rapid expansion of individuals paying small amount of money, $100, $200, to private companies to genotype their saliva. So 23andMe, I've just got a couple figures to show the level of growth of human biological samples they have. So 2015, about 800,000. A few months ago, 5 million. Ancestry DNA, a few months ago, over 7 million. Last Thanksgiving, when Ancestry DNA had not last month, a year ago, last month, when Ancestry DNA had a sale over Thanksgiving, in one weekend they sold 1.5 million tests. I have a note here that that's 2,000 gallons of saliva. That's a modest-sized pool in one weekend. The third company, Family Tree DNA, which you've probably heard less about 1 million a year ago. So when you just combine those, 3 private companies, you have about 1 in 20 of all U.S. Residents. None of that is possible if human DNA sequencing costs $10,000 each, $100,000 each. That's possible when it's $100 each, when it's $50 each sample. Here's some projections from just a couple years ago, 2015. This is the line of number of human genomes that had been sequenced from 2000, 2010, 2015 when these projections were made, so this is already dated. This is business as usual is the red line. If we just keep on going in the direction that had happened in the last year up through 2017, up through 2015, the projection was that in 2025, 2 billion human genomes would be sequenced. We're on pace, that's much faster than that. As this last slide, 1.5 million tests over a weekend is a different pace altogether. And these lines represent not only private companies, but also governments. So here's an example of governments around the world getting into this game of sampling large numbers of their populations. So enrollment to date is, so for example, Decode in Iceland, they have everybody. Iceland's not a very large country. Everybody is sequenced in Iceland. Here's one particular health system in one part of our country, in Pennsylvania. A couple years ago, they had over 200,000. You go to the United Kingdom Biobank, over 500,000. Kaiser Permanente, mostly in California, over 250,000. You start adding up, and those are lots of people. And there's a lot to suggest in the media that countries are not slowing down, they're speeding up. So this is from last year, Finland, 500,000 Finns, biggest biobank in the southern hemisphere in New South Wales, 3 million was their projection from last year, so that's what they're making progress on. And then you go to the US. Many of you might have heard of All of Us. There's a local chapter of All of Us here collecting human DNA. The effort here is 1 million. As you recall from a few slides ago, 1 million samples is what Ancestry DNA got in one weekend. We're going to spend 10 years to do that here through the US government, through the federal government. So, technological improvements. One thing that they've led to is the amassing of enormous samples of human DNA, as I was just describing. So that on its own might not have a lot of implications, but when you pair that with my next point, then we're going to get to some policy implications. So my next point is statistical improvements. What are companies, what are governments, what are researchers doing with these data? And they're aligning these huge data sets, this big data, with statistical improvements from computer science, statistics, econometrics, and so on, to do things that weren't possible before in terms of predicting, based only on DNA, people's outcomes. So how do they do this? I'll just give a short explanation. You amass, so you do what's called a genome-wide association study, which is a scan of everyone's DNA and asking, so you take a data set like 23andMe has, 5 million people, and you put it all in one data set, and you throw a lot of data mining techniques to scan for places in the genome where individuals seem to have differential outcomes. And by places, I mean you do these scans over 10,000, 100,000 to millions of statistical analysis. These are our chromosomes. You line up all the letters that are inside these chromosomes and go letter by letter. If you have an A versus a T there, are you more likely to do X? Are you more likely to be tall, more likely to have blue eyes, more likely to be have cardiovascular disease? And you do that in every location where there's variation across the genome. So you have tens of thousands, in fact millions of statistical tests. And you can only do that many tests when you have so many millions of samples in the background. So you do this big scan, statistical scan, and in the end, what you want to do is you want to visualize all your findings. And I'm going to describe what this is in just a minute. But this is a set of findings, so there's hundreds of thousands of potential findings here, and you try to do a scan of them and see where in the genome there seems to be a hotspot, a place where there's variation in the genome that seems to be related to an important outcome, cardiovascular disease, height, any sort of health outcome, but even broader than that. And then you take those findings and you try to make predictions. So let me build on these cartoons with some examples. So here is an example of a genome-wide association study on a particular outcome. The outcome is here. This is, in layman's terms, this is whether or not you report a funny smell in urine for someone who just had asparagus. Okay, and so what is done here is that in every point in the genome, the question is asked, does an A, a T, a C, a G here, is it related to someone's report of do I smell urine in that particular way? And what you see is nothing except right here. So this is a very, I think, clear picture of an example where there's a clear genetic association, a clear part of the genome that is clearly mapped onto that one outcome and nowhere else. So this is a simple case where there's maybe a very simple genetic architecture to this phenomenon. Okay, so are we happy with what is presented here? Underneath, there's hundreds of thousands of statistical tests. The fact that none of them have a peak that's very high means there's nothing there, but except there's one place in the genome, this huge spike where people are different in their reports on this smelling capability. Okay, and this is called a Manhattan plot, and what you're looking for are tall buildings. And here we have one skyscraper, and the rest little apartments. Any questions about that? Because I'm going to now add on a couple more examples. Okay, so this is a simple case. Slightly more complicated would be egg allergies. So you see a huge spike, but also some potential spikes here. But more generally, there's just a lot more taller buildings than here, than the last. Here, one single skyscraper. Here you have a single skyscraper and a lot of moderate skyscrapers in egg allergy. And similarly, milk allergy, up here's food allergy and combined. Peanut allergy is this upper right, lower left, egg allergy, lower right, milk allergy. And what visually I think what this should be getting across is that these outcomes, these traits are more genetically complicated. There's tall buildings across the genome versus this one, simple.
Question?
Yes?
Does the word genome refer to the formula to make a single person or the whole human race?
The question was, am I using genome to refer to a single person versus the whole human race? I'm not sure if I can answer that question. So I'm referring to genome as so let me be clear. What I mean here is that when investigators look at every single location of these 4 letters, they're asking, at this location, is there a difference between food allergy? If not, what about the next location? What about the next location? What about the next location? So that's what's visually plotted here, is a scan across every location in the genome, which is the collection of all the 3 billion letters I've been describing and more. So I think the answer to your question is that genome is more than what I'm saying. I'm describing I'm describing these 3 billion locations. There's more variation in humans, but I guess I want to repeat that in all these 3 billion locations, 99.5+% of them, meaning almost all of them, are exactly the same in everyone here and everyone who's ever lived. I just want to read. So we're looking at places where people differ, which are in the millions of the 3 billion possibilities. Okay, so this is slightly more complicated in the sense that there's more areas. So it's across more chromosomes you see taller buildings, and there's not one single site that says, yes, you have cow's milk allergy, or you don't. There's many, so we call this polygenic, many genes. This outcome, polygenic versus single gene. Yes?
Does this correspond to the SNPs?
Yes. So the question is a technical one, which is what you call these, and these are called single nucleotide polymorphisms, or SNPs. That's a long way of saying a single letter change, single nucleotide polymorphism, single letter change. So it's in areas in our genetics where there is variation. Yes.
The sites of the bigger spikes are the same in all 4.
Almost. So here,
I mean, the highest ones are all in the same.
so the observation here is that on chromosome 5 there appears to be a tall building in each case. And that would suggest then to geneticists that there might be a common cause genetically of these 4 outcomes, that there might be a genetic link between them. But it's also true that there are buildings that differ across them, so that it's not that there's one SNP, to use this language. There's not a single letter variation that is the thing that tells you whether you have a food allergy or not, in the way that there is a single letter variation that says you have sickle cell or not, as I was describing a few slides ago. Yes, in the back.
Are seasonal or pollen allergies involved in this?
So the question is about, what about environmental influences? That's right. So this is meant to showcase genetic influences not related to environments. So actually a lot of my research is about how genes interact with their environments. This is not what's meant to be what is trying to be shown here. What's trying to be shown here is that regardless of what environment you are in, pooling data, taking data from many parts of the country, from many parts of the world, in fact, are there common buildings, skyscrapers, in people across many places? So it's meant to be examining genetic signals that are it's not presenting environmental signals, which are very important. But the exercise here is to examine genetic signals. So these are still somewhat simple. I mean, they're polygenic. Many genes are affecting the outcome. And these are all, I think, what you'd expect is what's going on in genetics, which is to hunt down genes related to health conditions. Okay, so let me build on that point a little bit, which is to say you have all this information, and say you're a clinician, and I say, well, here's all the areas in the genome that seem to make a difference in egg allergies. Here you go, clinician, what are you going to do with that? So what's one thing that is done, so that's a very complicated problem. You have all these areas that could matter. So what's happening now is to take summaries of all this information and to create a single score, like a credit score, where a credit score is a lot of information about the bills that you've paid or not paid over time and so on, and you have a single number. There's something very similar happening on the genetic side, where all these results are pooled together, pushed together into a single index of risk called a polygenic score. So it's for traits that are polygenic, meaning many genes affect the trait, and we're going to get a single score that then clinicians might use in their office setting to say, I have a population that has been genotyped, meaning that someone has, you walk in the clinic, you've already contributed your DNA to the hospital. Say the hospital has run has run all your letters, figured out which letters you have, and you can then enter the clinic, and the clinician could say, I've got your DNA. Based on that, you're a high score or a low score for cardiovascular disease, say. And we're going to change your treatment, or we're going to go in this direction versus an alternative direction based on your genetic score. So this is now maybe what you'd expect. It's under the general framework of precision medicine, which is combining genetic information about your health and health risks into the clinic and making treatment decisions based on that risk. So it might be exactly what you'd think, which is we're going to use all this information to predict your likelihood of having a cardiovascular disease of some sort, and that's going to be useful in the clinic. So all that, the way that's working now in the clinic is to generate a single score, which is a genome-wide across the whole genome, taking all the information that's from these type of figures. So when you walk in, you would have a high score versus low score, like I said, a credit score. So in the population, these scores are going to be bell-shaped. Most people have modest risk or no risk. Some people have very low. Some people have very high risk for many traits. So most of that's probably not so surprising. This is a figure of just how many of these genome-wide association studies have happened in the last 10 years. And the answer is over 3,000. So approximately, you'll see a new genome-wide association study released every day. So it's just a huge number of new studies, and this figure, 39,000, is the figure that now stops 2 years ago. So a lot has happened in the last 2 years, like I was saying about the book. But in the last 10 years, there's been almost 40,000 instances where a single letter has been tied to a health trait across these 3,000 genome-wide association studies. And this number is also growing quite a lot. Okay, so now we're going to make a big shift, which is I think you know what's happening on the health side. Now the same technologies, which are taking biological samples, meaning saliva, figuring out what letters you have, and then predicting an outcome is now being used in other non-health settings. So that's where I'm going now. So again, this is dated 2 years ago, May of 2016. We have our first Manhattan plot, which you're familiar with. You're looking for big spikes, big buildings, skyscrapers. In this case, the skyscrapers are telling you where in the genome is related to your years of schooling, how far in school you went. And in a study 2 years ago, there were 74 locations in the genome. There were 74 skyscrapers where if you had an A versus a T, a C versus a G, it actually was predictive of whether you went to college or not. Yeah. So the same tools that you were probably maybe somewhat comfortable with or seemed like it would be obvious to do on egg allergies, cardiovascular risk, are being ported over to other outcomes like years of schooling. And all the same tools apply. It's a scan all the way through the genome, A's versus T's, and we're going to make a predictive measure on years of schooling and ask, what are we going to do with that type of predicted measure? So like I said, this is 2 years ago. It was on 300,000 people. 2 months ago, we have the new version of this, which is on 1 million people, and there are now 1,200 built spikes. There are 1,200 skyscrapers linked to whether you went to college or not, or your years of schooling.
Question?
Yes. So the question is the comment is that there's a difference between years of schooling captured here and intelligence. Exactly. No argument there. So keep that in mind. This is just an analysis of something that you could do, which is, can I predict years of schooling based only on your A's and C's and T's and G's?
Can you predict past years of schooling?
These are all people who are 50 years old, 60 years old. This is past years. This is completed years of schooling on over 1 million people in many countries, not just the United States. Okay.
So define predictive.
I'm going to do that in just a minute. So this is not telling you the predict. This is saying not how each of these predict education. It's saying that there is a signal. And in fact, here there are almost 1,300 separate signals across the genome that is related to how far in school you go. So there's just like the cardiovascular disease example in the clinician's office, where you could just come in and have a risk polygenic score of how likely you are to have cardiovascular disease. You can do exactly that for years of schooling. And in fact, here I'm doing another shift. I'm now outside the clinician's office. I, as an individual who have had my DNA genotype from 23andMe, 23andMe sends you back your information, the A's and C's and T's and G's. And I, as an individual, can go to this website this is from researchers at Columbia and NYU, and upload my DNA, and I can get my polygenic score for years of schooling. Just like the cardiovascular polygenic score, I can get my education polygenic score. And to answer the question before, what's the level of prediction on the genetic side? It's growing a lot in the last 5 years. And right now, a single score based only on A's and C's and T's and G's is as predictive as the other single most highly predictive score for your education. So let me unravel that sentence a minute. What do we think, if we only knew one thing about somebody, what do you think is the single most predictive measure of your schooling? Just one, you only get one measure. So I'm hearing money, intelligence, all those are, those are great guesses. And they're right in range of the single most predictive. But in fact, it is family history. It's how far in school your mom went. Right? So, and that's going to, if you only know one thing, like everything that you said is true. If I knew your income, if I knew your IQ, that's all predictive. But if the question is, if I only know one thing, what's going to be that measure that's most predictive, and it's your mom's schooling. Now, this measure is becoming to be competitive with that mother's schooling. It's being competitive with family history. So knowing only one thing, it's still true. Mother's education is the core prediction, but this is running a close second. And this one's going to get better, because as more people get their DNA sequenced, these measures are getting better. Okay, yeah.
Is there a problem with the samples you're using?
So the question is there a problem with the samples I'm using? That's a very deep question, actually. So let me describe one.
People with more education, more apt to get their DNA.
And then the question is it true that people who are more highly educated actually contribute their DNA to these studies? All that is true. In general, that's true, but the samples underneath these predictions are now becoming representative samples. So, for example, the UK Biobank is 500,000 Brits who are nationally representative of that country, of that nation, and that's not the only case. So, for example, 23andMe is highly non-representative. It is a highly white, educated, rich people in 23andMe. So that's not a representative sample. So these are good concerns, and I haven't said the word non-white yet. Other ancestries, and that's a key issue going forward, in that every one of these predictions are predictions made off samples of only individuals of European ancestry. And the figure, I say the figure is 90% of every data set is represented, of all the people in all of these data sets are represented by people of European ancestry. So I'm sure we'll come back to that. There's a lot of questions that come from that issue. So like I said, this is outside the clinic. This is me. I can upload this on my laptop and get my score, as anyone else here could. So we're really decoupling the clinic from these type of measures. And let me just make that point. Yeah, go ahead.
When you said you would be able to upload your genome, is it really just SNPs? that you get from
So the question is, what exactly are you uploading? And right now, or I should say maybe a year ago, it is SNPs. That's a technological, that will be a change in technology where eventually it will be your whole genome. Yes, so right now it's just your SNPs. Eventually it'll be your whole genome, every letter. So let me maybe make one more clarification. So measures of these single locations are not typically every location in your genome. Like I said before, 99.5% of them are the same across everyone, so there's no variation in those. But there are places where 1 in 1,000 people or 1 in a million people might have a different letter, and those type of places in the genome are not covered very well in the 23andMe. So that's the question of what exactly are you measuring. There are places in our genome where so-called rare variations, places where there's been a mutation that maybe affects 1 in a million or rare, that's not covered very well in the 23andMe type technology. But that's a temporary issue. Whole genome sequencing is becoming so cheap that, meaning you get every letter, all rare variants and so on, that this is a temporary issue, and that's actually part of the point that I was making earlier, is that these scores are going to be more and more predictive, not less predictive, as some of that information is backfilled. Here's an example of a private company, outside your clinic, offering for $199 screening for breast cancer genes. So my point here is decoupling the clinic, decoupling your relationships and your healthcare provider with the information in your genomes. And many companies are getting into this act of direct-to-consumer genetic predictions. So give me your data, and I'll tell you what wine you like.
What wine?
This is unlock insights about nutrition, fitness, even the wine you might prefer.
Better than we can ourselves.
So the point is better than we can ourselves. That's a great point to keep in mind. There's as you move outside the clinic, regulations to what companies can promise weaken. What's the evidence base that they can provide really good wine recommendations? Zero. Like they have no evidence that's true. That's true. If they were, so here's another example. So Spotify is an online service to set up what you're listening to on your iPhone or on your computer. Here's one of their latest ads. Spotify now wants to get your DNA so that they can recommend what your playlists are. My point more, so this is all junk. There's no great evidence that Spotify or the last company can do what they're promising with any accuracy. But my point about policy, which I'll be coming back to often, is that there's no regulation here that says you cannot do this. So let me dig into policy a little bit more, which is that while the wine preference and your playlists might be fun, there's some deeper issues with what's going to be done with this information, or what's possible to be done. And how I frame this in general is that we have new opportunities to discriminate. And it's moving from, and where the big idea here is that it's moving from age-based discrimination, sex-based discrimination, race-based discrimination. Those are groups of people that you discriminate against, old versus young, or Southerners versus Northerners. Those are groups. We can now discriminate based on particular people, based on their genomes. And I want to now walk you through some examples and where we have regulations in place, and mostly where we don't have regulations in place. So where we do have regulations in place is all comes from what's called GINA, Genetic Information Non-Discrimination Act from 2008. And it applies to two areas in our lives: health insurance and employment. Those are the two areas that it's illegal to be discriminated against based on your genetics. Full stop. There's no other areas in your life where it's explicitly illegal. So long-term care insurance, car insurance, home insurance, all legal to discriminate against on genetics. And I put this up here because this is a fragile law. It's been on the books for 10 years. It was almost overturned twice in the last 2 years, once by a single vote, all wrapped up in repeal and replace efforts of Obamacare, of ACA. So this was hanging by a vote of employers being able to discriminate against people based on their genome, based on their genetic information. Point 2, what about other parts of our lives, of our social lives? So I started this slide about a year and a half ago, and here's my co-author here making the point of what happens not when Spotify merges with 23andMe to dictate what playlists, music playlists you might have, but what if dating sites merge with 23andMe to then part of individuals' account being your genetic scores for education, but more importantly, maybe your genetic scores for Alzheimer's disease. How is that going to change what matches you have in these type of online dating markets when a person reveals that you have a very high likelihood of having Parkinson's or depression? And that was all speculation a year and a half ago. Here's 2 companies doing exactly this right now. So Gene Partner, and here's the app version of Pheromore here, which is give me your DNA and I'll match your DNA with my customer base for part of the matching criteria. So, and again, this is maybe to restate it, there's no regulation against anything like this. There's no regulation against potential romantic partners discriminating against others based on their DNA. Okay, so point 3 goes back to the beginning of the talk, which is what else might we be very interested in predicting? And let me remind you that you have had the letters in your genome your whole life. You had them as a fetus, and you'll have them the rest of your life. So what else might we want to predict based on using these same tools that we can make on education and on cardiovascular disease? And so then the next set of questions, again, unregulated questions, are what do we actually want to know about fetuses or babies? Do we want to be passing on to potential parents the sex of the child, how early, eye color of the child? Some of the eye color, hair color, those are incredibly easy genetic predictions to make. Height, the accuracy on height is something like 80%. Weight, more complicated questions of athleticism, IQ, going back to this point, we can certainly predict education. The genetic signals on education are very highly correlated with IQ. So what do we want? What information do we want before the baby's even born? Would we even want to know facial features of the baby, predicted facial features? Do we have any desire? So we've had this desire for a long time. Here is the Dr. Seuss with his patented creation of what is your baby going to look like. His technology was really different. His technology was to take 2 photographs, overlay them, and here's what your child's going to look like. So a lot of baby girl children predictions had mustaches, right? So the accuracy, not as good. What about when the accuracy is very high? So this is a paper from over a year and a half ago using only DNA, A's and C's and T's and G's to predict facial features. And I'll just remind you that, so these are all adults, so we know what the truth is of what their faces look like. The accuracy here, though, is high and will be getting higher. And the point here is that these A's and C's and T's and G's are, of course, exactly those genetic markers that you had as a fetus. So this is what you can create exactly these types of faces for fetuses. What is this fetus going to look like when they're 30? Yes.
Explain this a little bit more. I don't understand what you're predicting.
We are comparing actual pictures of people decontextualized from their ears and their hair color and so on with DNA predictions of the facial features of people.
From their DNA?
Just from their DNA. And the fact that these look similar is, and this is 1.5 years ago, the technology is much more accurate now. But again, to repeat, you can create these faces based on a sample from a fetus. Okay, so, and that's a larger question of, like, what do we want to know about fetuses is what sorts of discrimination will we allow about our future generations, those who have not been born yet? So the technology already exists, as many of you know, that you can take a biopsy of a single cell at day 3 and take all this genetic information and then do whatever you want. So right now what we do is we screen for genetic conditions, Down syndrome, for example. Right, so that's just standard practice. But the same A's and C's and T's and G's can be used to do this at day 3 because your A's and C's and T's and G's don't change. And this is just to quickly say that other countries are way ahead of us on this and we face no regulations. Other countries face no regulations and a lot of excitement about doing this. So this is, one point, this technology is growing 5 times faster in China than the United States about this pre-implantation genetic screening. Yes. Yeah, I will. So the question is, who owns the genetic data when you submit your data to 23andMe? And when you read, was there a clarification? So that was the question on hand, because the larger question is about privacy and who owns these data. So that's going to vary depending on what country you're living in. If you're living in the United States, you're going to be signing away your right to these data.
You sign away when you pay for it.
You're going to spit in a little bottle, and you're going to send the data in, and you're going to sign and pay for that service. And when you do those steps in the United States, 23andMe will, I think it was just last week that they signed a deal with GlaxoSmithKline for $300 million to share their DNA data with one of the drug companies, right? So the short answer is, if you're in the United States, you are explicitly signing your right from that information. Now they're going to send it when you submit it and pay for it. Now they're going to send it back to you. So there's a partial ownership here. You still own it, but you now it's actually difficult to cease and desist out of these data sets. Yes. So the question now is, what about why can't this be anonymous? I want you to hold that, because in principle, it could be. Yeah, I know. So the question is, why is this not anonymous? Why can it not be anonymous? And I'm going to answer that in just a minute, because the answer is very much deeply about what this genetic information is telling the companies about you and your family and your biological relatives. Okay, so make sure I answer that, though, because this is a very important question. This slide is just to say that what I'm describing is the direction that things are going is higher levels of predictability, more traits. We're not at the very end of this type of analysis. Each of those companies are continuing to enroll millions of people each year. They're asking the enrollees of their services, 23andMe, AncestryDNA.com, and so on, are asking more of their participants in surveys and so on. So the direction here is more genetic prediction, not less. And there's other directions I'm going to describe in a minute. So the last point is that there's both a part of this analysis that's very much on the health side. It's very much on the precision medicine. It's very much how can we optimize treatment of conditions that are important based on your genotype. But in much of the same way those analyses are done, Other analyses can be done and those are the years of schooling type analyses I'm describing, the facial features. It's the same set of statistical tools that are doing both. And there's no explicit regulations on what can be done, what traits can be predicted based on genetics. So coming to privacy. Who can obtain and act on genetic information? And here's just a slide. This slide is meant to ask, well, what do we want the answer to be? Because right now the answer on privacy is who cannot discriminate against you is employers and health insurers. And like I said, that's been a fragile law that was almost overturned twice in the last 2 years. But the rest of the list, there's no law against your schools using genetic information to make placements in math and reading. They're not doing that necessarily, but it's not because the law is stopping them. It's not because there's been an affirmative case that we do not want genetic information used in admissions decisions to UW. Why not? There's a lot of prediction in these A's and C's and T's and G's that might be related to how likely you are to complete your degree, which degree maybe you should go into. Those might be very useful, and right now, UW, I'm sure, is not using this information, but it's not because there's a legal restriction. Should there be a legal restriction? Other issues about access to technologies, meaning as the predictions get better, who will have access to them? It does cost money to have your genome sequenced. Is it going to be up to you to pay that cost, or is it going to be covered by insurance? What about people who don't have insurance? Are they going to be left out of any types of precision medicine advances? Privacy should really be a big deal here because I think that we should anticipate a massive hacking of a company like 23andMe. Not that I have any information here, but any inside information here, but just longer-term history, credit card company was hacked, 143 million customers. Just 2 weeks ago, Marriott revealed 500 million customers, right, with names, addresses, passport information, credit card numbers. And that sounds very much of an invasion of privacy. Let me just describe that a hack on 23andMe would be worse than that. Why would it be worse than that? Your current address, your credit card number, even your social security number, you can change all those. You can change your name if you wanted to. You cannot change your DNA. Point two, if your DNA is hacked, it's both permanent, like I said, you can't change it, and it's predictive about you. Your DNA being hacked is different than your credit card number being hacked in terms of what sort of information it gives the public about your risk of Alzheimer's disease, which is not something you can change. So the hack on 23andMe, or something like that, is going to be a different order of magnitude of what is revealed and what is predictive about the people who have contributed their DNA. And let me go back to this point of who in our society cannot use that information. And health insurers and employers. That's all. Everyone else could. Long-term care providers. They can use your status of Alzheimer's disease prediction to discriminate against you. And so this is more related to your point. Just a couple weeks ago, the journal Science investigators asked the question, if there was some sort of hack, if there was sort of public release of this information, who could we find with this information that already exists? Their first, so this is identifying information on just genetics. And so their first cut is that we could match almost 2/3 of every person in the country that we would search for, we could find them in this data, not because they're directly in genetic data. Most of you have not had your genetics tested, but your family could have, right? And when your family submits their DNA, they're submitting your DNA too. When your biological family submits their DNA, not when your spouse submits their DNA. When your biological relatives submit their DNA, they're submitting your DNA too. And this is the more chilling point in their abstract. The technique they were describing, they could find, what they're saying in the near future, they could find nearly any U.S. individual of European descent in the data. Meaning, almost anyone they would search for, they could find, because all they need is a third cousin or closer match. And the fact that we're so related, all of us are, and so similar, the likelihood that you have a third cousin in one of these data sets or closer is very high. So privacy here, this is a little bit more of an answer to your question. Why can't you be anonymous? And this is part of the answer. And the point, the comment, which I agree with, is there's no regulation of in what instances can that information be used against you. The regulation is health insurance and employment, and it's not everything else. The second point is while you're legally protected, to enact that protection, you need to go to court, which is also, so there's 2 points. There's a whole set of our life that's not protected at all, and that which is protected, how you get protection is to go to court after you've been, after you've lost, that's right, after you've lost your job or whatever it is. Yes. So the comment is, even though companies will say it's protected, and I'm sure they do their best to protect it, that it only requires one instance where it's released, because your DNA doesn't change, right? If your credit card number is released today, you have one remedy is to get a different credit card number, right? To get a different credit card. You do not have such a remedy for your DNA release. That is your DNA, and it's your family's DNA. If your DNA is released and it has information about Alzheimer's disease, that is relevant for your children, because some of that information is passed on to your children, right? It's relevant for your other biological relatives. That's telling a company something about them in their biological risk. And so a couple ongoing, so I've already mentioned this is one example from 6 months ago, that pre-existing conditions is related here, because many pre-existing conditions are strongly genetically predicted. So there's some interaction between what we know on the genetic side and what we're going to call pre-existing conditions. Let me say it a different way. We are at a point where we could start thinking about conditions that are genetically predicted before they emerge, and whether we want to cover those. So right now, a pre-existing condition has to have emerged to be pre-existing. On the genetic side, you can imagine at least thinking about some conditions that are very likely to emerge, and having some protection over those conditions before they emerge. So this is in the suite of, while we're weakening pre-existing condition protections, in the future we'd also be weakening genetically based pre-existing conditions. And here's another avenue that employers are kind of trying to scoot around this, the GINA law, which is that employers cannot discriminate, which is, there's a growing set of employers that are providing for free genetic testing for their employers, for their employees, for their workers, to test for heart conditions and so on, and provide that quote-unquote service to them. But at the same time, the employers get some of that information back. So there have been ways around even some of the GINA law. This is also outside the clinic. It's not about breast cancer risk. In this case, it's 190 diseases in your newborn's genetic code that, I think in this case, it costs $650. So this is just to put a point on, what do we want to know? What do we want to regulate about the information, the genetic information taken from newborns? So here it's not because the technology isn't there that it's not possible. We want to ask whether we want this type of testing to be done direct to consumer, where there's no clinical handshake. So I'm going to stop here because I want to allow more time Q&A. What this slide is describing is that I do have some slides on gene editing if we do want to spend some time on that in Q&A, but we don't have to. We can certainly stick to just genetic prediction and some of the laws. So let me walk through. I'm going to skip some of this and say I can come back to it, and formally finish by saying that UW is out in front here, I think. So we're currently have candidates coming in right now on social genomics. This is a cluster initiative from the central campus, from the chancellor's office, from the provost's office, from WRF. Many of you know these names. This is millions of dollars that these entities have invested in UW being a world leader in this area. In one of these three positions is going to be on someone whose main work is on thinking about policy and social implications of all what we've been talking about today. So I just want to highlight that UW is very much out in front here in realizing this is a really important issue. I'll acknowledge a lot of sources of funding that Millard already outlined, and I'll thank him for doing that. So I've been very well supported here at UW and in private foundations. And I'll formally say thank you and have contact information, and then we'll have a Q&A. So thank you.
Questions?
Okay.
I'm hearing questions. I'm happy to do questions right now. Go ahead. So the question is, you've just described cases, you've described some findings that genes are related to years of schooling in college. What about the environment? Totally fair question. And I mean, one reaction is that both are important, and my own view is that the environment is much more important than individual genetic factors, and that there's interactions between genes and environment in predicting how well people do in school and how far they go. This is clearly only part of the story. Totally acknowledge every one of those points. I think the point that I want to just focus on is what are we going to do with the genetic information that is predictive of years of schooling in our lives, even if it is not, and I agree that it is not the full story, and even the majority story. The fact that it's part of the story in years of schooling, what do we want to do about that? I think so I acknowledge each of those points, and those are very well taken and exactly correct. I'm just carving out a small slice of that and asking, now that we're able to predict years of schooling like we've never been able to do before, and whether you go to college or not, what are we going to do with that information, and what are we going to not allow to be done with that information? Thank you for the question. Yes? So two-part question. I might have to ask you to remind me of the second one so the first one is gene editing. What do we do? What can we do now? What's possible? And with a particular case of Huntington's disease, which is one of these single gene disorders that's quite important. The second question is, what can we do when we get this information? What kind of environmental changes could we make about nutrition, what we're eating, are we exercising? So let me come back to that. So gene editing first. Many of you saw headlines recently about Chinese scientists gene editing fetuses, which is to say just that it's rapidly growing. It's very quickly moved from a year or two ago being just off the table. We'll never gene edit human embryos to, okay, we will, but it has to be about therapeutic purpose. So there's two ways of gene editing. There's one to take away a mutation that's detrimental, a therapeutic use, and there would be another one where you would insert a mutation that might be advantageous. That is, I just have such a colorful slide here. I'll put that in the background. So this is gene editing where we're trying to get new basketball stars or new track stars. This is from over a year ago. So the science is just rapidly expanding, and this is a little bit technical, but I want to just make the point that there's two different types. There's two purposes of gene editing, even between therapeutic and advantageous, which is, are you editing an embryo? Are you editing someone who's been born? Let me just say it that way. So it's a human, fully formed human. And why that's important is that an edit on an embryo, where you change a letter, that proliferates across all new cells and is what's called a germline edit, meaning that embryo will pass along that edit to that embryo's children. Okay, so that's one type of edit, but it must be early on embryonic, so it gets proliferated through the body. A different edit is you take a fully formed human who is likely to have Huntington's, they have the Huntington's gene, and you edit their cells And now the problem is we have 1 trillion cells. We need to edit all of them. Right? So what happens in practice is that scientists are trying to find where in the body Huntington's is actually expressed in a detrimental way and edit those cells. And that's a one-time thing, meaning that when you have your cells edited for Huntington's, I mean, this is not successful yet. Well, there's one case where it seems like it was successful in Britain. When you have your cells edited for Huntington's, you still pass down to your offspring the Huntington's gene. After birth. Because there's a difference between editing the first cell when you're an embryo and all future cells as you become a fully formed human have that edit versus taking a human that has 1 trillion cells and trying to edit out the mutation. And the second one's just much more complicated. And in fact, we can't do that yet. The first one is what was happening in this recent example in China, where you edit an embryo and try to make that efficacious. Second one, I'll be more quick about, which is if you get information, what can you act on in your daily life about nutrition and diet? And I think that's some of the promise. I mean, going back to this idea of precision medicine, it's in that umbrella of how do genes interact with the environment. When you have a high genetic load for Alzheimer's, is it going to be, are you going to lower your risk by having a different job, eating differently? This is all much more in standard scientific inquiry. And we don't have, I'm going to say we have very limited answers now that a person getting their DNA back could act on in terms of Alzheimer's, Parkinson's. The umbrella term would be lifestyle changes that you can make. I think there's very few and not very much evidence among the few of what one can do. And that goes to the question of why do we give this information back and so on, like if you can't act on it. Yes. So the question is, does this information raise the possibility that we have an ethical obligation of genotyping everybody and targeting resources towards people who might have disadvantages based on some of their letters in their genome? I don't have an answer for that. That's precisely the type of question I think we'd want the public and our legislative bodies to be deeply considering. Because it is an implication of, do we have an ethical obligation of, especially when we can imperfectly predict these things, right? So we're going to be making mistakes on prediction. But are we ethically bound to genotype young children, say at birth, for dyslexia, because there might be interventions one could do before we see that they have problems reading. Way before that, there might be interventions one could do. Some people, that sounds like something potentially useful to do, but it opens the door to when would you shut the door? In what dimensions of our genetic code would you say that's off limits? We cannot intervene or maybe even create genetic measures on other outcomes that aren't dyslexia, because the same analysis that would give you a prediction of dyslexia could also give you a prediction of IQ or facial features for that matter, and how are we going to differentially allocate resources to people who have different genetic backgrounds in that way? Excellent question.
The idea that biology is destiny for a long time. I don't think we should embrace it.
Yeah. So the comment, which I agree with, is that we shouldn't, none of this should be pointing us to this common claim in the past that biology is destiny. I certainly think that's right. These are predictions, just like your credit score is a prediction. It's not destiny. In very limited cases is a destiny. Huntington's, maybe there's some destiny, but almost everything else is not. Eye color is probably destiny. There's almost, in every other case, but that then goes to this other point. When you know it's not destiny, do we want to intervene and reshape the possibilities that people might have based on their genetic code? Yes. So the point is the umbrella point is we know so little about what these genes are actually doing. And how can we act on information where we have such little knowledge of what's actually going on in the genome? Which is one question about the use of genetic predictions in IVF clinics, which are completely possible. Right, you have a set of embryos that have been generated. You can genotype each of them and select which embryo you want based on polygenic scores for education or IQ or whatever else. None of that's illegal. I'm not saying that's happening. It is happening in China, I guess. It's not clear it's happening here. But what are the other implications of doing that? I mean, one that is commonly discussed is if people select only on measures of IQ that will have... it's relatively well known that selecting on measures of IQ also increase the risk of schizophrenia. So that's an add-on effect. That's an effect that you weren't maybe desiring or anticipating, but something like that is very much a question. And what that leads me to, though, is what policy, what laws do we want to have enacted about these choices? Right now, it's a free-for-all. It's based on individual decision-making in which country you're in and what clinician you have about what types of choices that you can make. What kinds of guardrails do we want to put on those decisions? And we've done it twice with health insurance and employment. There's currently nothing on the IVF clinic side about what can and can't be measured from embryos. Do we want any guardrails there? These are questions. I don't have an answer to that. But your point is even a bigger point about when we do these types of selections, there might be quite negative, unanticipated consequences because we know so little about what the genes are actually doing. In the back. So the question is a question about what limitations other countries might be putting on this type of technology that we might consider. So we have varieties of countries that are moving in quite different directions, where a country like China, I think, is very much interested in exploiting this technology to its fullest. So I'll put that aside to say that there are countries that are quite aggressive in these technologies, but other countries in Scandinavia, I think, in particular, are much more. They seem to be more interested in putting regulations on the genetic screening of embryos and fetuses, of what information can be gathered. That's one aspect. But I'm afraid I don't have any examples in mind where there's a country that's way ahead of us in the anti-discrimination side of things. Maybe someone else actually in the room has an idea of where there's other aspects of life where genetic information cannot be used. Part of the issue is that, for example, our healthcare system is just so different than every other country, as you know, that so a lot of the questions we have about paying for care are just not at all related in some countries. So our regulation about health insurance cannot discriminate on genetic information based on price. It's just immaterial in a lot of countries because they don't have any prices. It's just nationally insured population, so there's no such thing. So there's a little bit of that about us being so different in some parts of our life to begin with, that we just have a lot of our life that has the possibility of needing regulation, like long-term care and car insurance, house insurance. All those insurance mechanisms can discriminate. For example, it's a great question. I need to think more about examples. I don't have many in mind. So the answer, and I should have added that, is that even on the 23andMe side, if you're a citizen of the EU, you can ask for your data to be deleted from 23andMe, and it must be done, or is at least legally bound to be done. You still have to sue probably if it's not done. But we as U.S. citizens don't have that right now, whereas if you're submitting your DNA from France, I think you can say to 23andMe, take me completely out of your data, and that must be done. At least legally it must be done, yes. Thank you for that example. The question is, do we have existing groups interested in this and promoting policies? I don't want to say the answer is no, but are there national groups that have a large voice? That's harder to see. I don't have examples of that in the United States case of a group that already has a platform that this is what should be done. This is part of the point of talking. I'm presuming that some fraction of you, this is news, and I think that that's going to be true across the country. There's some news in this presentation about what's being done with DNA, the privacy issues, and that part of the answer is that I'd imagine that most people wouldn't know that this is an issue to think about. And at the same time you have that, the technology is just so fast that we're already 10 years behind probably, like now. So unfortunately, I don't have a more optimistic stance of a website I can just point you to and say that's how you get involved. I think part of this discussion is the startings of being involved in the back. The question is, to what extent are the predictions accurate across traits? Wide range of accuracies. So the direction is towards getting more accurate predictions because more and more people are contributing their DNA, so the data sets are getting larger and larger and larger. But that's really important. So that's in some sense the place to be optimistic, is that a lot of the individual predictions aren't very good right now. So it doesn't stop companies from discriminating against you necessarily, but they're not going to be able to do a very good job of it right now. So maybe it's still time to get in place some guardrails about, as these predictions get better and better, what companies, what areas of our lives do we not want those predictions to be used for judgments about us. So to answer your question, so there's a spectrum. Those traits that we think are as genetic disorders caused by a small number of mutations, those are highly predictive, as I'm sure you know. As you move towards the social science type outcomes like years of schooling, they get much less predictive. But I think that doesn't rescue us entirely from whether we want even those noisy predictions used for us or against us in different settings outside of health insurance and employment. Yes. There is Louise was making there is a group, should I repeat the group?
Electronic Privacy Information Center.
EPIC, which you might be able to remember. The other point is, I'm sure there's other instances of policy being slow versus technology being very fast. I think this is one of them. So if we would have done this discussion 2 or 3 years ago, it would have been different. It would have been different. In a lot of these slides wouldn't have even been available. And why I say that is because if we do it again in 2 or 3 years, it's hard to even speculate how different this kind of what's known of what's been done in the background, both in scientific endeavors, but also from drug development and the widespread release or the even wider spread collection of human DNA. Because one thing that will happen, this is still a speculation, but I think it's likely to be true, is that there will be a move from relatively low bits of information, these SNPs, these letters across the genome that are much less accurate when we try to predict outcomes, to having hundreds of thousands and millions of people doing whole genome sequencing. That will be happening here, but also all around the world, because the technology is just changing prices so dramatically. Everything's getting so cheap that people are promoting, even in education research, it's cheaper to genotype children and create a lot of measures of their health and other risk scores than ask a 10-page survey question of their parents. It's just because it costs $20 to genotype them, and you get thousands of trait predictions if you want them, versus the bread and butter of older survey research, where you ask people about their lives and you ask their parents about the children and so on, and that's very expensive to do, whereas it's so cheap to genotype them. So it's just I don't see any breaks here. I see just full steam ahead, many more people being genotyped, and then wanting to think about, as many of you brought up, well, what kind of guardrails do we want, and then what are those first steps? The first steps are partly you all being interested and asking your own legislators, what are your stances on this? Or is there something, if it's something that you're really interested in doing? Yeah, in the back. So the question is law enforcement taking advantage, is using this information? And really the backdrop of some of this privacy and science information is the large and growing number of cold cases that are being prosecuted because of DNA. So it's in the case, there's dozens now of old murders and rape cases from 3 decades ago that are now being prosecuted. And so far, it's never that the assailant themselves were genotyped. They didn't have their DNA in 23andMe or in the law. It was that their third cousin did. So when the DNA specimen was at the crime scene, that was matched to the third cousin of the criminal. And that has been, like I said, dozens of high-profile cases recently. So this is not a clear, this is a good example of where do we want law enforcement involved in these, because these are privacy questions. They knew with a lot of clarity that the person whose biological specimen was at the crime scene was the third cousin of this person in the data set. And so that limits the range of who's possible, or even the second cousin, or the brother of that person. That really limits. It doesn't say for sure it's you. They have to go get a warrant and get the potential criminal's genotype done. But again, this is a very gray area of the law of privacy, and there's been a lot of benefit here of old cases being solved just because of changes in technology and the amassing of these huge data sets. So I think we'll probably be a little conflicted with how much we want to shut down those and maybe give up some privacy so that more of these prosecutions can go forward. I'm speaking for myself. I'm sure there's a variety of opinions just in the room. Yes. I don't know if it's different than what I'm describing with these serial killer arrests, which is to say that it's never the killer themselves that have been genotyped into a biobank, it's their relative. And you find this match that then limits who the possible people are that actually left the biological specimen. So that's the main part of law. Oh, actually, there's one other area that's pretty interesting. There was a case in New York City, I think 2 years ago, where there was a murder, and only part, I'm going to say it was, let's say that they found a severed foot. So there was a murder, and they didn't know who it was, but they used the DNA of the foot to create a face of the person, and they eventually solved the crime. Is that something we want to bar? So this is a question where there's, I think, not easy answers, especially when it comes to law enforcement, because again, there's privacy concerns here. This person didn't consent to any of that, and so on. So the point was that Promega is the kit provider. They're providing the little cups that you spit in, but otherwise aren't so involved. First. Right. So the point is that the proportion of cases that are cold cases and are stranger cases is small. The question is, how do we value the solving of those cases when we can so easily maybe do it with DNA, but otherwise the resources are real. The difference between having a detective on the case for 10 years versus just using the biological specimen and going to a biobank, those are quite differences in cost and in prosecution, which is of interest.
I think we have to thank Jason and let him go because he has an executive committee meeting that he has to be at.
I do.
Soon. So let me say thank you very much for a most enlightening talk. You don't get much out of the rather chintzy organization that we belong to, but we do want to express our gratitude to you. This is
Thank you all.
a travel blanket and pillow,
Yes.
which we hope you'll enjoy in
I
your
will.
travels. Thank you very much.
Thank you.